What if somewhere in the inky depths of our oceans, hidden from sight, there existed creatures that could cheat time itself? Imagine a world where the ticking clock of aging, so familiar to humans, is not a universal law but a negotiable rule. In this shadowy underwater realm, the promise of immortality doesn't belong to myth or legend but to real, living animals. Today, science reveals that some ocean dwellers defy the normal limits of life, pushing the boundaries of what it means to age and eventually die. It's tempting to picture these creatures as eerie or ghostly, lurking in the blue abyss, timeless and unchanged. Could the ocean hold the answers to the secrets of eternal life, or perhaps the dark consequences of it? As we begin this journey, prepare to question the very nature of life cycles and mortality as we've always known them. Aging is often thought of as an inevitable march toward decay. Wrinkled skin, slowing bodies, and ultimately death. But what really causes aging beneath the surface? At its core, aging is a complex biological process linked to the gradual deterioration of cells. DNA damage accumulates, vital molecules become less efficient, and cells lose their ability to regenerate. Yet some marine animals seem to sidestep or vastly slow down this process. The cold, deep, and mysterious ocean environment itself acts like a natural time capsule, where low temperatures and scarce resources compel creatures to adopt survival strategies that stretch their lifespans far beyond what seems possible on land. Could aging be less about the passing of years and more about the environment and evolutionary design? This radical idea challenges our deepest assumptions. One of the most astonishing oceanic immortals is a tiny creature hardly larger than a fingernail. The immortal jellyfish, scientifically known as Turritopsis dohrnii. Its life cycle reads like a sci-fi thriller. After reaching adulthood and reproducing, rather than succumbing to the ravages of age or death, this jellyfish can actually reverse its aging process. It essentially rewinds its biological clock by transforming its adult form back into its juvenile stage, a polyp, which then grows anew into a fresh jellyfish. This process, called transdifferentiation, is like a biological reset button at the cellular level, where mature cells change identity to start a new life. Imagine a creature that, whenever threatened or damaged, doesn't just repair itself but goes back to the very beginning of its life. It's a mysterious power almost unimaginable and hidden in plain sight under the ocean's waves. But this biological rewind is not invincibility. In nature, the immortal jellyfish faces countless dangers: predators, disease, changing water conditions. Although its cells can evade the aging process, the jellyfish is never completely safe from death by external forces. Still, its ability to repeatedly restart its life cycle offers a glimpse into a world where death is less certain and life's end may be postponed indefinitely, at least on a cellular scale. What if this remarkable ability could someday unlock clues for human medicine? The implications stretch beyond marine biology into the realms of genetics, aging research, and even philosophy. Our own mortality might one day be viewed through the prism of these tiny, pulsing creatures that refuse to grow old. This phenomenon raises haunting questions. What would it truly mean to live forever? Would eternal youth be a blessing or an endless sentence? The immortal jellyfish's looping existence feels almost like a curse, trapped in cyclical rebirth with no final release. Unlike humans, who experience time linearly, these jellyfish could experience a kind of biological stasis where life is an unending loop. In the quiet, dark ocean depths, a creature flickers in and out of existence, forever young yet never quite free. It turns the comforting idea of life's progression on its head and invites us to look at aging and death in a profoundly different light, filled with both wonder and unease. Encounters with such creatures also ignite curiosity about the ocean's hidden corners and other marine animals that might hold their own secrets of longevity and arrested aging. Beyond the jellyfish, some species of sharks live for centuries, deep-sea sponges thrive for millennia, and lobsters possess biochemical tools that let them defy traditional aging processes. These creatures hint at evolutionary experiments with lifespan that stretch far beyond human experience. Could their biology contain keys that open doors to longer, healthier lives for all animals? Or are these adaptations simply survival mechanisms shaped by the ocean's extreme conditions? The ocean's depths are a realm where time bends in strange ways, and where life itself is a mysterious, shifting puzzle. Yet, as we stand in awe of these seemingly immortal beings, there's a creeping sense of mystery, and even horror, to consider. Immortality in the ocean may come with invisible costs or limitations we don't yet understand. Does living forever trap a creature in endless cycles of survival devoid of growth or new experience? Does it make them vulnerable to stagnation, eternal witnesses to a changing world? The beauty of these animals is tangled with a darker, almost gothic story of existence beyond time's reach. Their fragile forms pulse silently in watery shadows, reminding us that the unknown depths guard both miracles and ancient enigmas we have barely begun to comprehend. Our journey starts here, at the edge of known science and deep human curiosity. The promise of immortality underwater is not just a fascinating biological anomaly, but a profound invitation to rethink life, death, and what lies beyond. Could the answers to humanity's oldest questions be waiting in the dark tide, beneath currents filled with creatures that challenge fate itself? Where science meets mystery, our exploration of oceanic immortals will lead us further into realms where life may not end, but only transform again and again, forever. Stay with the mystery, as the ocean's secrets slowly unfold. Aging is something everyone fears, yet few truly understand its hidden mechanics beneath the surface of life. We see it as the slow, relentless march toward frailty, wrinkles, and eventual death. But what if aging isn't simply a steady countdown but a complex web of biological events, frequent repairs, and inevitable compromises? At the core, aging results from the gradual accumulation of cellular damage. DNA strands break, proteins fold incorrectly, and critical molecules falter. Our bodies try desperately to patch up these microscopic wounds, but over time, the effort becomes overwhelming and systems begin to fail. Where does this process begin, and why does it seem so inexorably written into our fate? The ocean holds some of the oldest, most enigmatic creatures on Earth, offering clues to how aging might be slowed, paused, or even reversed. Many marine animals dwell in conditions uniquely suited to longevity. Cold temperatures reduce metabolic rates, slowing the chemical reactions that can cause cellular damage. The crushing pressure of deep waters also limits oxygen availability, which paradoxically protects cells from oxidative stress, one of aging's main drivers. As a result, species like the Greenland shark can live for centuries, with some estimates suggesting lifespans of over 400 years. Glass sponges, rooted to the ocean floor, seem to defy time as they grow at incredibly slow rates, surviving for thousands of years in near-pristine environments. These creatures are living time capsules, their bodies telling stories far older than human civilization. The ocean's extreme environments force a biological bargain: slow down life's dance or face collapse. But slowing aging is only one part of the story. At the cellular level, the secret to longevity often lies in how organisms maintain their DNA. Cells contain telomeres, protective caps at the ends of chromosomes that shorten each time a cell divides. When they become too short, a cell can no longer replicate and effectively dies, contributing to tissue aging. Some animals, like lobsters and certain turtles, produce an enzyme called telomerase that rebuilds telomeres, granting their cells an extended youth. Unlike us, these animals sustain telomerase production well into adulthood, keeping their DNA intact and cellular machinery youthful. This remarkable ability hints that longevity might be linked to a biological toolkit, one that humans possess but do not fully utilize in adult life. Could unlocking this toolkit in humans someday halt aging itself? The phenomenon of cellular renewal doesn't stop at telomeres. Some species possess extraordinary regenerative capabilities. The axolotl, a freshwater salamander, can rebuild entire limbs and organs, an almost magical demonstration of cellular plasticity. Even some marine animals can regenerate damaged tissues, fighting off the toll of time like microscopic immortals. At the heart of these feats are stem cells, master builders capable of transforming into a variety of tissues. In certain invertebrates, like the freshwater hydra, a gene known as FOXO plays a role in keeping stem cells eternally youthful, preventing the body from succumbing to age. Yet the exact interplay between genetics and environment remains shrouded in mystery, a frontier that continues to puzzle scientists as they uncover the ocean's biological secrets. In the silent blue depths, where sunlight barely penetrates, life takes on an eerie quality. Creatures that defy aging exist alongside fragile, ephemeral beings, all coexisting in a delicate balance. The ocean is a strange paradox where immense lifespans compete with brutal, sudden death by predation or environmental shifts. Biological immortality in marine creatures doesn't mean immunity from external threats. Disease, predators, and habitat changes still pose deadly challenges. The mythical immortal jellyfish, for example, can theoretically reset its life cycle endlessly but remains vulnerable in the wild. This frailty wrapped in mysterious power creates a shadowy tension. Immortality as biological resilience paired with existential vulnerability. Might these creatures' paradoxical lives hint at the true nature of aging? A dance between internal potential and external fate? Scientists have long been captivated by these paradoxes. Research into marine longevity has expanded our understanding of aging from a fatal destiny to a complex, malleable process. The regenerative mechanisms discovered in ocean species are inspiring breakthroughs in medical science, including potential therapies for age-related diseases and tissue engineering. These insights force a reconsideration of aging as a biological constant, suggesting instead a spectrum of possibilities determined by genetics, environment, and chance. The ocean's hidden laboratories offer models of life resilience, perhaps even keys to human longevity. But even as we hope to apply these lessons, the mysteries and limits of aging remain deeply entangled with the natural rhythms of life and death. There is also a poetic, almost melancholic dimension to these findings. The creatures that avoid aging live in isolation, in extreme, unchanging environments far from the bustling surface world. Their timeless existence often seems like a stasis rather than true freedom. Aging may be the price we pay for change, adaptation, and the rich tapestry of life experiences. Living forever might trap a creature in repetitive cycles, preventing evolution or new growth, a biological limbo beneath the waves. What haunting balance does nature strike between life's continuity and its inevitable end? Could the fear of aging lie not just in death but in the loss of meaningful transformation? Understanding aging through the lens of these extraordinary marine animals challenges us to think beyond human limits. It opens the door to a future where age may no longer be a sentence but a condition that can be managed or even reversed. Yet, the ocean's deep mysteries remind us that the quest for immortality is riddled with uncertainties and paradoxes. As science delves into the biology of longevity, it uncovers not only knowledge but profound questions about existence itself. The ocean, vast and unknowable, invites us to continue exploring the delicate line between life and the shadows of time. Among the countless mysteries of the ocean, few are as captivating and unsettling as the life cycle of the immortal jellyfish, Turritopsis dohrnii. This tiny creature, barely a few millimeters wide, has shocked scientists by displaying a remarkable ability. It can reverse its own aging process and essentially start its life over. Unlike most animals that follow a strict one-way trajectory from birth through aging to death, this jellyfish possesses a biological escape hatch, a process called transdifferentiation. When faced with stress, injury, or the natural wear of age, it abandons its adult medusa form and transforms back into its juvenile polyp stage, effectively hitting the reset button on life itself. Imagine a creature that, instead of dying, rewinds the clock to become young again and begins its life anew, potentially repeating this cycle indefinitely. The life of Turritopsis begins like many other jellyfish. Adult medusae, drifting gracefully through the ocean currents, release eggs and sperm into the water. Fertilized eggs develop into free-swimming planula larvae, which eventually settle on a surface and grow into polyps, tiny, colonial animals that bud off new jellyfish. Normally, these medusae mature, reproduce, and then perish, as is typical for jellyfish species. But the immortal jellyfish breaks this mold. When threatened by environmental hardships, such as starvation, physical damage, or illness, it can escape death by reverting to the polyp stage through a process that fundamentally rewires its cells. This reverse metamorphosis is unique in the animal kingdom and makes Turritopsis dohrnii one of the few known biologically immortal species. The key to this regeneration lies in transdifferentiation, where specialized adult cells dissolve their identities and transform into a new, undifferentiated mass of stem cells. From this cellular blank slate, new polyp cells emerge, allowing the jellyfish to rebirth itself fully. Unlike typical regeneration where lost limbs or tissues might grow back, Turritopsis does something even more profound. It moves backward in its entire life cycle. This process can theoretically continue infinitely, making the jellyfish biologically immortal in laboratory conditions. However, in the wild, most individual jellyfish likely fall prey to predators or disease before they can cycle back, reminding us that survival requires more than just biological tricks. The transition from medusa back to polyp takes place over the course of a few days. As the jellyfish's bell contracts and its tentacles retract, it sinks to the ocean floor and forms a cyst-like structure. Within hours, the cells of this cyst begin their transformation through transdifferentiation. Observations in captivity have confirmed this astonishing reversal, with some colonies renewing themselves multiple times over several years. Yet, the rarity of witnessing this process in nature adds a ghostly layer to the phenomenon. The immortal jellyfish's ability to live forever exists mainly out of sight, hidden in the depths or masked by its tiny size, a secret whispered only to the patient observer. Beyond the science, the life cycle of Turritopsis dohrnii poses profound philosophical questions. What does it mean for an organism to escape death? And at what cost? This perpetual rebirth might mean endless cycles trapped in a biological loop without growth or the release that death brings. The jellyfish's existence challenges the comforting narrative that life follows a linear path, filled with clear stages of youth, maturity, and death. Instead, this creature inhabits a shadow realm where time folds back on itself, where death is not an end but a detour. Is such a life a blessing or a form of eternal stagnation? Scientists continue to study the molecular mechanisms behind the immortal jellyfish's rejuvenation. Recent research has revealed that genes related to DNA repair, stem cell renewal, and cellular resurrection play crucial roles. Some of these genes show remarkable activity during the cyst stage, enabling the jellyfish to heal and regenerate damaged tissues effectively. These discoveries have spurred interest in the potential medical applications of understanding immortality, from fighting human aging to improving regenerative therapies. The jellyfish's life cycle may hold clues not only about animal biology but also about the fundamental limits of living systems. Despite its name, the jellyfish's immortality is not invincible. Predation remains a constant threat, and environmental changes can still prove fatal. Unlike fairy tales where immortality is absolute, here it is conditional, fragile, and partly hidden by nature's dangers. Yet, this fragile immortality, a blend of biological magic and harsh reality, is what makes Turritopsis dohrnii so compelling. It hovers on the threshold between life and death, constantly negotiating existence. The deep ocean waters it inhabits are its sanctuary and prison, a realm removed from human time but shaped by forces just as brutal and mysterious. As we peer further into the ocean's depths and the immortal jellyfish's enigmatic life, we glimpse a broader truth. Life and death are deeply intertwined in ways that defy simple understanding. The jellyfish invites us to reconsider our own mortality and the nature of time. Could the ocean's secrets one day unlock the path to immortality for other species, including ourselves? Or does this biological looping come with hidden costs we have yet to understand? The immortal jellyfish might live forever, but it also reminds us that the boundary between life and death is a delicate, shifting frontier, one of the ocean's darkest and most beautiful mysteries. In the mysterious realm of regeneration, the axolotl stands as a living enigma, a creature whose ability to rebuild lost limbs and organs defies everything we understand about biological limits. Unlike most vertebrates, which heal wounds only to scar and deform, the axolotl can restore entire body parts with astonishing accuracy, rebuilding complex structures like spinal cords, limbs, and even parts of its brain. This remarkable feat begins at the cellular level, where a unique population of stem cells springs into action following injury. These cells, unlike ordinary ones, do not merely patch up damage but orchestrate a symphony of regeneration, a process both intricate and eerily efficient. How does this aquatic salamander activate such extraordinary cellular machinery? And what secrets does this hold for the future of medicine and perhaps even immortality itself? The key to axolotl regeneration lies in the formation of the blastema, a cluster of progenitor cells that gathers at the site of injury. These cells act like master builders, proliferating and differentiating into the specialized tissues needed to replace missing parts. What makes the blastema so fascinating and unlike anything seen in humans is that mature, specialized cells nearby can revert to a more primitive stem-like state to join the regeneration effort. This cellular rewinding defies common biological rules, essentially allowing the axolotl's body to create a fresh start from damaged or lost structures. But how do these cells know exactly what to regenerate and where? Scientists have discovered that signaling pathways and chemical gradients within the limb precisely guide cellular behavior, ensuring new limbs grow with perfect shape and function. One signaling pathway crucial to this process involves retinoic acid, which creates a chemical gradient along the length of the limb. The concentration of retinoic acid near the shoulder is much higher than near the wrist, and this gradient acts as a positional map for regenerating cells. In simple terms, the cells read their location along this gradient and know whether to regrow an entire arm or just a hand. This molecular GPS guides the blastema to rebuild limbs with astonishing fidelity, a feat that remains impossible in humans. Such precision hints at a masterful control of cellular identity, and research is ongoing to understand how this positional code is established and maintained during regeneration. Neural stem cells within the spinal cord also play a vital role in the regeneration process, especially when the axolotl recovers from severe injuries. Incredibly, these stem cells synchronize their activity over surprisingly long distances to orchestrate tissue repair. When the spinal cord is damaged, signals spread like waves along the cord, activating stem cells to divide rapidly and replace lost neurons. This coordinated response happens within days of injury, revealing a complex communication network unseen in most animals. The factors triggering this synchronization are still being unraveled, but early studies suggest a mysterious signaling molecule acts as a messenger, instructing stem cells to mobilize. Understanding this process could revolutionize treatments for spinal cord injuries in humans, a tantalizing glimpse into the power of regenerative biology. Beyond signaling, epigenetic regulation, the chemical modifications to DNA and histones that control gene expression profoundly affects the axolotl's regenerative abilities. Unlike mammals, axolotls can activate or silence genes needed for regeneration without triggering scarring, a common roadblock in human healing. Key enzymes change the structure of chromatin, making regeneration genes accessible precisely when needed. Additionally, microRNAs, small molecules that fine-tune gene expression, regulate cellular fate during regeneration, ensuring cells proliferate, migrate, and differentiate in a controlled manner. This epigenetic orchestra ensures that regeneration proceeds smoothly and accurately, avoiding the errors that often occur in damaged human tissues. Could learning to manipulate these mechanisms one day allow us to tap into similar regenerative powers? The axolotl's ability to regenerate its limbs is not just about restoring form but recapturing full function. Regenerated limbs move with nearly identical strength and responsiveness, a testament to the creature's evolutionary mastery. This implies that the regenerating tissues do more than grow back. They reestablish nerves, muscles, bones, and blood vessels in perfect harmony. Studies show that nerve signals are essential for the blastema's growth. Without nerve input, regeneration stalls. This dependence highlights the deep integration between the nervous system and cellular regeneration, pointing to complex crosstalk that sustains life. The axolotl's story raises haunting questions. If lost parts can be perfectly replaced, what stops this process from continuing indefinitely? What are its natural limits? The regenerative magic of axolotls comes with intrinsic mysteries and paradoxes. Their remarkable healing requires a tightly controlled process of cellular dedifferentiation and proliferation, which if poorly regulated could lead to cancer-like growths. That these animals avoid such fate suggests evolutionary fine-tuning that balances regeneration with cellular control. In their quiet freshwater habitats, far removed from the dangers of ocean predators, axolotls glide through life with a ghostly grace, creatures seemingly encased in a time loop of restoration and renewal. Their uneasily perfect healing challenges our understanding of mortality, blurring the line between life, death, and rebirth in ways that stir both awe and existential dread. What secrets might the axolotl unlock for humanity? Could understanding its regenerative symphony pave the way for therapies that restore lost limbs or heal spinal injuries? Or even hint at deeper possibilities of biological immortality? Scientists remain cautious yet hopeful, as the complex signaling networks and genetic mechanisms involved are only beginning to be understood. One day, perhaps, we might learn to activate our own blastema, awakening dormant regenerative potential within us. Until then, the axolotl remains a living mystery, a delicate balance of biology, evolution, and hidden power, silently regenerating beneath the waters, waiting to share its secrets with the future. The axolotl's capacity to regenerate its brain is a phenomenon that pushes the boundaries of what we consider possible in vertebrate biology. Unlike humans and most mammals, whose brain cells rarely regenerate after injury, axolotls possess an extraordinary ability to repair damaged brain tissue, including the complex structures of the telencephalon, the front portion responsible for memory, cognition, and sensory input. This incredible power to regrow diverse neuron types is orchestrated by ependymoglial cells, specialized neural stem cells that awaken after injury to produce new neurons and replace lost cells. The regenerative process is far from spontaneous. It follows a highly coordinated sequence where injured areas are repaired, new neurons differentiate, and connections are gradually restored. However, even with this astonishing regrowth, the axolotl's brain regeneration has its limits, sparking questions about the true extent and functional recovery possible in these creatures. When the brain suffers injury, ependymoglial cells near the damaged tissue activate an injury-specific state, characterized by gene expressions linked to wound healing and cell migration. These cells then proliferate rapidly, spreading beyond the immediate damage site to ensure broad regeneration. Magnetic resonance imaging, MRI, has allowed scientists to observe this regeneration in vivo, revealing that wounds close within several weeks, and new neurons begin to repopulate the area shortly after. Intriguingly, the brain's response is not isolated to the injury, but involves a more global pattern, where distant uninjured regions also contribute to regeneration. Such systemic coordination suggests a deep, underlying network of signals guiding repair and reconstruction, creating a biological chorus that rejuvenates the brain in stages. The regenerative neurons in the axolotl's brain do not just fill gaps. They assume specific molecular identities matching the various neuron subtypes lost to injury. These newborn neurons acquire mature electrophysiological properties, firing electrical signals and responding to local inputs much like normal cells. They embed themselves within local circuits, restoring some degree of function. However, despite the impressive regrowth, the axolotl struggles to fully recreate the original long-distance axonal connections and fine tissue architecture that existed before injury. This partial restoration challenges assumptions that brain regeneration can perfectly reverse damage, hinting instead at a complex balance between regrowth and tissue limitations. The regenerated brain may look whole but remains subtly altered beneath its surface. One striking example of this regeneration is how the axolotl recovers neural function in the dorsal pallium, which corresponds to important brain regions for sensory processing. Localized injuries disrupt this area, yet the subsequent neuronal proliferation leads to significant new neurons appearing within weeks. Scientists traced these new cells using molecular markers, revealing increases in neuron subtypes crucial for brain function. Still, the regenerated neurons are organized differently, and long-range communication pathways are not fully reestablished. This architectural restructuring raises profound questions about how much brain function can truly be restored and what compromises regeneration entails. Can a brain rebuilt differently preserve memory, personality, and cognition, or does it become a ghostly echo of what once was? Ependymoglial cells, the neural stem cells critical to axolotl brain regeneration, exhibit behavior both remarkable and mysterious. These cells divide and differentiate to replace lost neurons after injury but do so within precise molecular programs involving gene regulatory networks. What triggers their activation remains under study, but it appears injury induces a unique transcriptional state distinguished by wound healing and cell migration genes. This state guides the cells through neurogenesis phases similar to those observed during normal brain maintenance, but in an injury-specific context. The fine line between activating regeneration and triggering uncontrolled growth is tightly controlled, and understanding this balance could unlock new therapeutic approaches for human brain injuries. The axolotl's neural regeneration extends also to spinal cord injuries, where specific populations of neurons in the brain become activated to promote repair in distant tissues. For example, certain glutamatergic neurons respond to tail amputation by stimulating axon extension into the regenerated tissue. They also induce hormone production in the hypothalamus to facilitate healing. This neural crosstalk between brain and spinal cord represents a complex systemic response to injury, emphasizing the integrative nature of regeneration in these animals. Such insights illuminate how neuronal networks orchestrate repair beyond localized damage and inspire hope for therapies that could harness similar pathways in humans. Despite their impressive capabilities, axolotls have limits to brain regeneration. Studies show that regenerated brain regions cannot perfectly duplicate original tissue architecture or long-distance connectivity. This limit reveals an important truth. Regeneration may restore structure and local function, but cannot entirely erase the past trauma's effects. The axolotl's brain, though remarkable, becomes a reshaped landscape where some functions recover while others remain altered or diminished. This imperfect restoration challenges the notion of biological immortality. Even creatures with incredible regenerative powers face intrinsic constraints and trade-offs. It serves as a humbling reminder that nature's mysteries are both wondrous and bounded. The mystery of axolotl brain regeneration invites us to rethink aging, injury, and the possibility of renewal. Could unlocking the secrets of neural regeneration lead to breakthroughs in human neurobiology? What if someday humans could awaken dormant regenerative programs to heal brain injuries or combat neurodegenerative diseases? Yet, the story also warns of the delicate balance needed to achieve such feats without unleashing chaotic growth or cancer. The axolotl, with its graceful dance between loss and renewal, embodies both hope and caution. Its uncanny ability to rebuild life after injury illuminates the boundary where biology meets the unknown, a frontier where science pursues the dream of healing beyond death's reach. Deep in the midnight zone, where sunlight never reaches and pressure crushes like an eternal embrace, life persists in forms that mock our understanding of time. Glass sponges, ancient sentinels of the abyss, cling to the seafloor, their porous bodies filtering nutrients from water that has circulated through Earth's oceans for millennia. Some specimens, dated by growth rings akin to tree trunks, span over 11,000 years, older than the pyramids, older than recorded history itself. These creatures exhibit negligible senescence, a state where aging markers like cellular deterioration barely register, allowing them to endure in perpetual slow motion. Crustaceans such as deep-sea lobsters and isopods scuttle across the darkness, their metabolism so sluggish that they evade the ravages of time, living centuries without the frailty we associate with age. What evolutionary sorcery enables these abyssal dwellers to whisper defiance against decay? And what horrors lurk in their unchanging vigil? The Midnight Zone, from 1,000 to 4,000 meters deep, crafts an environment hostile yet preservative. Temperatures hover near freezing, slashing metabolic rates and the oxidative damage that fuels aging on the surface. Scarce oxygen and nutrients force a thriftiness in energy use, channeling resources toward maintenance over growth or reproduction. Glass sponges thrive here, their siliceous skeletons growing mere millimeters per year, a glacial pace that accumulates no wear. Radiocarbon dating confirms individuals surpassing 10,000 years, their cells locked in a stasis where division rarely occurs, sidestepping telomere shortening and genetic errors. This realm's uniformity, unchanging cold, pressure, and dark, acts as a biological preservative, trapping life in amber-like suspension. Zombie worms, or ozdiacs, add a macabre twist to this deep time gallery. These slender invertebrates bore into whale bones that sink to the abyss, anchoring themselves for decades or centuries as they digest lipids from the skeleton. Females remain embedded lifelong, their bodies unchanging while laying eggs that drift to new carcasses. Negligible senescence keeps them viable across generations, their slow metabolism mirroring the sponges above. Enzymes within maintain cellular integrity, repairing DNA with eerie precision amid the bone's slow dissolution. Picture these worms as undead parasites, sustained by death's remnants in a cycle-defying temporal decay. Crustaceans of the deep, like the giant isopod Glypturus, embody armored longevity. Specimens reach ages of 70 years or more, their chitinous exoskeletons molting infrequently to conserve energy. Low temperatures preserve enzymes, preventing protein misfolding that plagues warmer climes. Telomerase activity persists, elongating telomeres and thwarting replicative senescence. These creatures evade predators through sheer endurance, outlasting threats in the void. Their eyes, reduced or absent, sense the world through chemoreception, navigating a realm where time dissolves into irrelevance. Protection from decay stems from multiple synergistic factors. Cold inhibits reactive oxygen species, primary culprits in cellular aging. High pressure stabilizes proteins, reducing denaturation risks. Nutrient scarcity selects for hyper-efficient repair mechanisms, like robust antioxidant systems and autophagy pathways that recycle damaged components. Glass sponges deploy spicules as both structure and defense, their slow growth minimizing mutation accumulation. Crustaceans hoard lipids, fueling longevity without constant foraging. This confluence crafts immortality not as active conquest but passive surrender to the abyss's embrace. Yet beneath the awe lies subtle horror. These immortals witness epochs unfold without participation. Glass sponges, fixed since the last ice age, endure continental drifts and mass extinctions, their awareness, if any, a silent scream across millennia. Zombie worms feast eternally on fleeting whale falls, their lives looped in necrotic symbiosis. Crustaceans patrol barren plains, unchanging amid cataclysms. Immortality here manifests as stagnation, evolution bypassed for mere persistence. Does sentience flicker in these ancient forms, trapped in biological limbo? Speculation creeps in. Could abyssal pressures forge true indestructibility? Future expeditions might reveal sponges harboring microbial symbionts that enhance repair, or worms with viral integrations granting resilience. Crustacean genomes, sequenced recently, hint at expanded longevity genes, ripe for human application. Yet tampering invites peril. What if transplanted mechanisms unleash uncontrolled growth? The deep's gifts carry shadows, promising renewal laced with the abyss's chill. As we ascend from these time-frozen depths, the question lingers. In a world demanding adaptation, is immortality evolution's cruel jest? Glass sponges, zombie worms, and crustaceans persist as living fossils, their negligible senescence a monument to endurance over progress. They challenge mortality's inevitability, yet evoke dread of endless, unchanging existence. The midnight zone guards these enigmas, veiling biology's outer limits in darkness. What other ancients await discovery, and at what philosophical cost?